pilot.
Real time.
Yes.
You will take a photograph and yeah.
It's real time or quantitative?
It's real time as well as quantitative.
Both.
Real.
Both.
Yes.
It's one machine or two?
It's one machine. It's always one machine.
I'm a
But
sociologist, so I don't know. But
okay,
our colleague
so
is,
I let you know that. Just come ahead. The difference.
video, yeah.
Difference in conventional PCR and real-time PCR is that in the real-time PCR here is a camera.
Yeah.
The lower part is likewise the conventional PCR.
Yeah.
Mean amplification is there. In conventional PCR it's same. But in this case we use a fluorescent dye. And here is the camera.
Okay.
That camera detects the presence of fluorescent dye. Because the signal of fluorescent dye, that is exactly proportional to the quantity of the amplified DNA.
Absolutely.
As much as is the fluorescent dye, that much would be the amplicons. That's why we
Right.
call it as real time. Means you can track the amplification at the same time as well as you can quantify what the amplicon or target is.
Taking
That's
the
the
sound?
basic difference. So the only
Focus
difference
on the
is
screen.
a
Yeah.
camera
Camera. Wow.
Yes.
Yeah. And the quantification.
and the chemistry as
And
well.
the chemistry. Yeah.
Yes, because in this case you don't need to go for
QPCR.
Yeah.
Because that's
Because-
what she wanted. Yeah.
in this case you don't need to go for what we call post PCR processing.
Right.
Mean you have to take out the samples and run on the gel. We can show you the gels as well. So this is the case of conventional PCR. Here these are conventional PCR.
Okay.
This one and video download. So whatever is the product, we run it on the gel. Then we are able to see the results.
Okay.
But in real time or QPCR, you don't need to go for this. You can directly take the results while your samples are within the tube.
So real time PCR and QPCR is the same.
Same.
And you have something called endpoint.
Endpoint
Yeah,
PCR.
you have that.
Both of these are called endpoint PCR.
Oh,
because
it's the same.
Yeah, these
so
are the
endpoint
terms.
PCR, quantity PCR.
Endpoint PCR is conventional PCR as well.
Oh, okay.
That's not.
Sorry, I'm also not a.
Okay. Real-time PCR, quantitative PCR, QPCR, these are all the same names for the same equipment or same
Okay.
technique you can
So
say.
that's a new one, or the endpoint is the old.
And the traditional one is called?
Conventional PCR we call it as, or you can say even endpoint PCR. Endpoint mean you can detect the results at the end of your experiment. But real-time mean you can detect the results as your reaction
And you
starts.
can quantify.
Yes, you can quantify as well.
Okay.
Yeah, quantification is important.
And this is biochemistry department.
This is biotechnology department.
Biotechnology.
Yes.
And also elaborate about this gel documentation.
Yes, this is gel documentation system. This is actually used. Bioride one. It's very good one. Here's the UV source. We put the gel here and just put on the UV light. You get the image of gel that whatever the DNA has been amplified or
Oh,
not.
you did like a photograph?
Yes, we can photograph it in the system.
Okay.
Because DNA can't be seen with the naked eye.
Of course.
Then we expose to the UV light. And the UV light and fluorescence. Where there is fluorescence, it means DNA is there. But again, in real-time PCR, all this is not needed. You can detect directly.
The problem with
So-
PCR,
so we can do the-
changes very frequently, you know, the technique.
No, it doesn't change very quickly. That
Okay.
is rather expensive.
Oh, that's
Nothing else.
So we can do the isolation and spreading of bacteria and DNA extraction.
Yes, we have that sort of system in the lab. It's we name it as being done because you can bombard your genes into the plant or animal source with the help of this gun.
Wow.
Because whatever is used, we coat DNA on gold particles and we put the tissue over here. Whatever is your target tissue, that may be animal or plant, and then we, with the help of that pressure generated through the cylinder, we push the button fire, and then you get penetrate your DNA into your target tissues. So if you want to carry some gene from the alien source, like from bacteria, fungi, or whatsoever gene you can incorporate. You might have heard about GMOs, development of GMOs, which means integration of foreign DNA into a cell. So this is
And gene editing is also.
gene editing is also done with the same. The basic difference is just in the chemistry. Gene gun is also used for the gene editing. So if you want to carry some gene, that may be new gene from edited gene, that all is carried through gene gun.
Okay, thank you.
Very much.
Yes, something.